Hello, all of you. I hope all of you are staying safe in this lockdown. In today's class, I would like to inform you about the mechanism of formation of ring structure of aldo and ketohexose. And under this, I would like to let you know about anomers, and after that, I will discuss about the formation of the ring structure of monosaccharide by Fisher and Haworth projection formula. And next I will discuss about the important property of cyclic monosaccharides, that is mutarotation. And in the last section I will uh, let you know something about the important reactions of carbohydrates, especially monosaccharides. So let us first discuss about the mechanism of the formation of ring structure of aldo and ketohexoses in especially in aqueous solution. So in aqueous solution, aldotetrosis, all monosaccharide with 5 or more carbon atoms in the backbone, occurs predominantly as cyclic or ring structures in which the carbonyl group has formed a covalent bond with oxygen of hydroxyl group along the chain. So in biological system, the monosaccharide generally exist in cyclic form, or you can say almost all the carbohydrates are generally exist in cyclic form. That's why this to know about the cyclic structure of monosaccharides or carbohydrates are very important for us. So monosaccharides that contain 5 or more carbon atoms generally forms cyclic structure in aqueous solution. So in aqueous solution, what happens? Monosaccharide often switches from acyclic form to cyclic form like this uh, through a nucleophilic addition reaction between carbonyl group and one of the hydroxyl group in the same molecule, right? And this reaction generally creates a ring structure of carbon atom closed by one bridging oxygen atom. So this is like this. So this is the so a ring structure of monosaccharide can be formed by two different formula. First is Fisher projection formula and second is Haworth projection formula. So first of all, I would like to discuss about the mechanism of ring structure formation in glucose in aqueous solution and the representation of this closed structure by Fisher projection formula. So this is the open structure of D-glucose, and this structure has aldehyde group. You can see here at C1 position it has aldehyde group, and it has been written as C double bond O and H, and in the sixth carbon there is hydroxyl group. So and in this structure you can easily observe that in the left hand side, in the right hand side, maximum hydroxyl group are present here, but in the left hand side only one hydroxyl group present at the C3 position, that is carbon number 3 position. Here at C1 oxygen is at C1 that is carbon number 1, oxygen is more electronegative, that's why it has partial negative charge, and carbon generally has partial positive charge. Similarly at carbon 5 position, oxygen has partial negative charge and hydrogen has partial positive charge. So due to these charge distribution, this oxygen interacts with this carbon atom and this hydrogen interacts with this oxygen atom. So as a result of that, ring structure of sorry, ring structure of alpha D glucose forms, right? And in this ring structure, in the place of carbonyl group, now there is one hydroxyl group, and this oxygen will be in this oxygen will be now in the ring form. So in this structure you can easily observe that here OH group is in OH group present, OH group bonded with carbon 1 is in the right hand side, and you can write this structure like this also, where this OH group present in the left hand side of the carbon number 1. So these two isomers which are differ in configuration around carbon 1 position are known as anomers, right? And these two structures, that is these two cyclic structure, are known as Fischer projection formula of D-glucose. So one if in one isomer OH group is in the left side of carbon number 1 position, then it is known as beta D-glucose, and if the same hydroxyl group present on the right hand side of the carbon 1 position, then it is known as alpha D-glucose, and both alpha and beta D-glucose are known as anomers, right? So now later on Haworth explain the ring structure of these two isomer based on pyranose ring structure. This is the pyranose ring structure, and pyranose is considered as the chemicals with 6 member consisting of 5 carbon ring and 1 oxygen atom. So this is the 5 carbon ring with 1 oxygen atom, and this is first carbon, this is second carbon, this is third, this is fourth, and this is fifth, and this is the oxygen atom. So Haworth structure are very easy to draw with the help of Fischer structure. So now I am going to tell you the excellent trick which is very useful to draw the Haworth projection, which is very easy to draw the cyclic structure of alpha D glucose on the basis of Haworth projection formula. So just concentrate on this structure of alpha D glucose, and here you will have to keep in mind one thing, that the group which are present on the left hand side should be at the top position, and the group which are present on the right hand side should be at the bottom position. So now I am going to write the structure of alpha D glucose on the basis of the Fischer projection formula of alpha D glucose. So first of all, you will write the structure of pyranose. So this is the structure of pyranose. Now you will have to draw the line like this. Just draw the line like this. Now give number to all the carbon atom present here. So this is the first carbon, this is the second carbon, this is third, this is fourth, and this is fifth. Now, as I said you earlier, that the group which are present on the left hand side should be at the top. So according to this, H is present at the left hand side, so it should be at this position. So H is here. Next, this H will be here, this OH will be here, this H will be here, and since at 6th position CH2OH is here, that's why this hydrogen should be placed at in this position, right? Now the group which are present and on the right hand side should be at the bottom. So hydroxyl group is in the right hand side, that's why it is present, it will be present at the bottom. Next is this hydroxyl group, next is this hydrogen present, next is this hydroxyl group, this is present here. So now students, this structure is, this cyclic structure generally represent which monosaccharide? So this generally represents alpha D glucose, and Now I would like to explain the formation of the cyclic structure of uh, beta D glucose on the basis of the Haworth projection formula. Again, for that, in beta D glucose, the hydroxyl group should be at the left hand side of the carbon number 1. So it is here. So that's why this is known as beta D glucose. And this cyclic structure is generally based on Fischer projection formula. So from this Fischer projection formula of beta D glucose, we will have to draw the structure of beta D glucose on the basis of Haworth projection formula, right? So for that, now again you will have to first draw the structure of pyranose. Just draw the line like this, right? And now give the number to all the carbon atom present here in this cyclic structure. This is first carbon, now this is second, this is third, this is fourth, this is fifth, and CH2OH is the sixth. Just write this CH2OH at the sixth position here. And now, as I told you earlier, that OH group, that all the group which are present in the left hand side of this structure should be placed at the top. So here first will come OH group. So OH group is here. Then will come this H group. Then next is this OH. Next is this H. And since CH2OH as the sixth position, that's why this H will be, we can write this H at the bottom. So this structure generally resembles the cyclic structure of beta D glucose on the basis of the Haworth projection formula. So in this case, in these cyclic forms of alpha D glucose, again in alpha D glucose, hydroxyl group is at the bottom, but in beta D glucose, the hydroxyl group at the top of the carbon 1 position. So that's why these two isomers are considered as anomers, and because these two differ in configuration around carbon 1 atom, right? So these two are considered as anomers. Now I would like to discuss about the trick for the formation of cyclic structure of fructose in aqueous solution, right? Sorry. So in practical, all carbohydrates present in cyclic form in living cells, tissue and organs. So as I told you earlier, here I will also let you know about the trick to draw Fischer and Haworth position formula of cyclic structure of fructose. So the molecular formula of fructose is C6H12O6. So let us see the open structure of fructose. This is the open structure of fructose. On the basis of its structure, it is found to contain a ketonic group at second carbon atom, and first carbon atom and sixth carbon atom have hydroxyl group, right? And third carbon atom contain OH group in the left hand side, and fourth and fifth carbon atom have OH group in the bonded with the OH group have OH group in the right hand side, right? Like glucose, fructose also exist in cyclic form. So let us see those structure. In this structure, CH2OH group has been written in the left hand side like this. Left hand side of the C2 carbon number 2 like this. This is the C2 carbon number 2, and CH2OH has been written in its left hand side. And see here, oxygen is more electronegative, that's why it has partial negative charge, and carbon has partial positive charge. Similarly, the hydroxyl group present at the carbon 5 position, oxygen has partial negative charge and hydrogen has partial positive charge. So due to these charge distribution, this hydrogen interacts with this oxygen and this oxygen interacts with this carbon, right? And due to these interactions, cyclic form of the fructose, uh, cyclic structure of fructose forms like this. So we can see here at C2 position a hydroxyl group forms as a result of the interaction between this hydrogen and this oxygen, like, and this oxygen is the part of this cyclic structure which also form the bridge between this carbon and carbon number 5 and carbon number 2, right? So in this way, through Fischer projection formula, we can write the cyclic structure of alpha D glucose. So here in alpha D glucose you can see that hydroxyl group present at the right hand side of the C2 carbon, right? And in you can write this structure like this also, in which hydroxyl group present in the left hand side of this carbon atom, that is C2 carbon atom. So the structure in which the hydroxyl group present in the right hand side of the C2 carbon atom are known as alpha D fructose, and the same hydroxyl group present in the left hand side, then that isomer of fructose are known as beta D glucose. And both these alpha D glucose and beta D glucose are considered as the anomers, because as I have already told you earlier that if two isomers differ in configuration around hemiacetal or hemiketal carbon atom are known as anomers. So here the C2 carbon number 2 is the hemiketal carbon atom. That's why and there is change in configuration of OH group in these two and C2 carbon atom in both these two isomers. That's why these two structures are these two isomers of D fructose are considered as anomers, like, right? Now this cyclic structure of of D fructose generally resembles the another cyclic carbon structure, that is furanose, which generally has 5 carbon rings, that is one generally has 5 carbon in its ring structure, that is first, second, third, fourth, and one will be here in the fifth, and with one oxygen atom, right? And so on the basis of this furanose structure we can write the Haworth projection formula for the cyclic structure of fructose. So this is the open structure of, this is the Fischer projection formula for alpha D fructose. Now similarly you will have to draw the structure of furanose ring, and again you will have to draw the lines like this, and now you will have to give number to all the carbons like 1, 2, 3, 4, 5 and 6. So at 6th position just write CH2OH like this. Now again the groups which are present in the left hand side of the of this structure should be placed at the top, right? So here CH2OH is there, so CH2OH should be written at the top. Now second would be OH, OH is here, next is H, H is here, and since CH2OH is the position of CH2OH is here, that's why you can write that this hydrogen atom at the bottom. Again in the right you will have to write all the, so the groups or atoms which are present on the right hand side of this structure should be placed at the bottom. So here OH group is in the right hand side, so that's why it is here. Then next will come H, then OH, and again H will be, H is already there, so you will not have to write. So what this structure resembles, so this structure resembles the cyclic structure of alpha D fructose on the basis of Haworth projection formula. So now, according to the Haworth projection formula, these two isomers are known as anomers, right? And these two isomers differ in the configuration around first carbon atom. So in alpha D glucose, the hydroxyl group should be placed at the bottom, and in bottom of the carbon 1 position, and in beta D glucose, the hydroxyl group should be at the top of the carbon 1 position. So since these two differ in configuration around carbon 1 position, or you can say hemiacetal carbon position, that's why these two isomers are known as anomers. And in the case of D fructose, in alpha D fructose, the hydroxyl group is present at the bottom of the hemiketal carbon atom. Here hemiketal carbon atom is carbon number 2, and the same hydroxyl group present at the top of the hemiketal carbon atom, and it is known as beta D fructose. So both alpha D and beta D fructose are the isomers of D fructose, and these two isomers are known as anomers of each other, because in these two isomers differ in configuration around hemiketal carbon atom, and here hemiketal carbon atom is carbon number 2. So here I would like to revise some of your basic concepts of chemistry. I think you would have know that from your higher secondary chemistry concept, that when hydroxyl group interact with aldehyde group, it forms hemiacetal, and when hydroxyl group interact with ketone group, then it forms hemiketal, right? So same concept has been applied here. In sugar also, in D glucose and D fructose also, you have seen that hydroxyl group react with aldehyde group in glucose, and again in fructose hydroxyl group interact with ketonic group. So same reaction same reaction is happening in the case of the cyclic formation of the glucose and fructose also, right? So this is all about the cyclic structure of glucose and fructose according to the Fischer projection formula and Haworth projection formula. So now I would like to describe about mutarotation, which is a specific property only. So mutarotation is the specific property of cyclic monosaccharide. So this is very important property of the cyclic monosaccharide. So you can, as I told you earlier, that it is a special property only specific to cyclic monosaccharide, and mutarotation comes from two words, that is, first is mutation and second is rotation. And mutation means change, and rotation means optical rotation, or you can say a specific rotation. So as I taught you earlier, that a specific rotation is the special property of optically active compound, in which this compound rotates or responsible for rotating the plane polarized light to a certain degrees of angle, right? So mutarotation can simply defined as a dynamic change in the rotation of plane polarized light as it passes through a solution of some sugars as an equilibrium mixture of alpha and beta D glucose is formed, right? So mutarotation, let's try to understand mutarotation with alpha D glucose and beta D glucose, right? Because both are optically active compounds and alpha D glucose are less stable than beta D glucose because of the uh, possibility of the repulsion between hydroxyl group at carbon number 1 and carbon number 4, uh, as at both the carbon atom they are present at the same site. But in case of beta D glucose, OH group at carbon number 1 and carbon number 4 are at opposite position. That's why the chances of repulsion between these two are quite less, and that's why beta D glucose are more stable than alpha D glucose. So if we have now if we have alpha D glucose and we dissolve that glucose in water, then some part will convert into beta D glucose, and after some time it will achieve the e- equilibrium, right? And equilibrium in solution, and if we will take beta D glucose, then reverse case will occur. Since alpha D glucose in an optically active compound, so its specific rotation value uh, is 112 degree, and specific rotation of beta D glucose is around 118.7 degrees. When it will achieve the equilibrium, so when it will achieve the equilibrium, then a specific rotation of equilibrium mixture would be only 52.4 degree. And if we are dissolving this alpha D glucose, it will convert into beta D glucose through this acyclic structure. This is the open structure, right? And the percentage of alpha D glucose will be 36%, and percentage of beta D glucose would be around 64%, and percentage of acyclic structure would be only around 0.02%. So if we have 100% of alpha D glucose in solution, then through this acyclic structure 64% of the alpha D glucose will convert into beta D glucose, and in reverse case, if we have 100% of the beta D glucose, then 100% of beta D glucose in one solution, then through acyclic structure 36% of the beta D glucose will convert into alpha D glucose, right? So we can say that at equilibrium the solution at equilibrium the equilibrium solution will have 36% of the alpha D glucose and 64% of the beta D glucose. So how can you how from these value, how can you calculate the specific rotation of equilibrium mixture? So in equilibrium mixture, what you have 36% of the alpha D glucose plus 64% of the beta D glucose, right? So in this calculation we are taking 36% of 112.2 degree and 64% of 18.7 degree, and when we will solve it, the value of the specific rotation of the equilibrium mixture will come around 52.4 degree, right? Since beta D glucose is more stable than alpha D glucose, so in equilibrium mixture the concentration of beta D glucose will be more in comparison to the alpha D glucose. So as I told you earlier, the mutarotation is the a specific property of of a cyclic monosaccharide. So we can say that in mutarotation, what is happening, uh, the rotation for alpha D glucose, that is the rotation of alpha D glucose, changes from 112.2 degrees to 52.4 degree, and a specific rotation of beta D glucose, that is 18.7 degree, changes to uh, 52.4 degrees. So in this way, this is the important property of cyclic monosaccharide, and this is all about mutarotation. Now I would like to discuss about the reaction of carbohydrate, especially monosaccharide. So to get an idea about what kind of chemical reaction will under is generally depends on what kind of functional group they have. As you know, most of the monosaccharide possess either an aldehyde or a ketone group. Hence the chemical reaction that are monosaccharides, hence the chemical reactions that a monosaccharides will undergo is depend on these functional group. These reaction are oxidation reaction. Through oxidation reaction, aldehyde and ketone group generally converts into acid, carboxylic acid group, right? And this is the way through which oxidation takes place in carbohydrate. In case of reduction reaction of carbohydrate, the aldehyde and ketone group converts into alcohol groups. And there is another one reaction, that is Oza-Jones test. In Oza-Jones test, we generally react the monosaccharide with phenyl hydrazine. So this is the special kind of monosaccharide test through which we can determine that which kind of monosaccharide we have in our sample. So let us discuss all these three reaction of monosaccharide one by one. So reaction of carbohydrate, that is or monosaccharide, that is oxidation. So in oxidation, what is happening? So in oxidation, this aldehyde group in presence of oxygen and enzyme glucose oxidase converts into a carboxylic group, and this is and by this way D-glucose has been converted into gluconic acid. There should be D-gluconic acid, and gluconic acid is gluconic acid generally occurs in fruits, honey and wine, and is used as a food additive, and which is known as acidity regulator, right? Now, so there is a chance of oxidation of both aldehyde and aldehyde group present at carbon 1 position, and hydroxyl group present at carbon number 6. So when this group will be oxidized, then it will form glucuronic acid. So glucuronic acid is generally produced in the liver of human, and it generally helps in transporting various hormones and drugs from one part of the body to another. So this is all about the oxidation of monosaccharides, especially glucose. Now, when aldehyde group present at the C1 position of the D-glucose reduced, it generally forms alcohol. And in D-glucose, when this reaction reaction happens, then D-glucose converts into D-sorbitol. So like that, like this way, other monosaccharide also reduce and form these kind of sugar alcohol. These are known as sugar alcohol. So when D-glucose oxidizes, it form reduce, then it form D-sorbitol. When D-galactose reduce, when the reduction reaction of D-galactose happens, then it will produce it produces D-dulcitol. Likewise, when D-ribose when the reduction of D-ribose happens, then it forms D-ribitol. So sorbitol and dulcitol, when accumulate in tissue in large amount, is generally cause strong osmotic effect leading to swelling of cells and certain pathological condition. That, for example, cataract, peripheral neuropathy, ma- like this. And mannitol is useful for reducing intercranial tension by forced diuresis. So just go through this and you will come to know about the different function of these sugar alcohols in biological system. So now uh, I would like to discuss about the Oza-Zone test, which is an important reaction of carbohydrates. So through this test, we generally identify the different kind of monosaccharides in our uh, sample. So this is the D-glucose. When it reacts with phenyl hydrogens, then it will form glucose ozazones. So ozazones are highly colored and crystalline compound and can be easily detected. Each sugar has a characteristic crystal form of ozazone. So by this way, you can identify different kinds of sugar by this reaction, because uh, by this reaction different sugar forms different kind of crystal structures. So example is maltose form petal shaped crystals. In the similarly lactose form powder puff shaped crystals. Galactose form rhombic plate shaped crystals. Glucose, fructose and mannose forms broomstick or needle shaped crystals. So by this way, if you got puff shaped crystals in your sample, right, then you can say that yeah, in my sample the lactose is present, right? So like this way, this ozone test used to identify different kind of monosaccharides in your samples. So this is all about today's class. So if you find any difficulty in understanding anything in today's class, so you are most welcome to contact me through phone, through WhatsApp or through mail. So thanks a lot to all of you and stay safe and bye bye.